Thank you. And let's find the slides and get the second day of the conference started. Hello, CDKL5 community everywhere, Canada and everywhere. I hope you had a great day yesterday with the International Awareness Day. And today we're kicking on the program, talking about all of the announcements and news from the last 12 months since the last patient conference about CDD treatments. My name is Anna, as Sangeeta explained, and I work with the LouLou Foundation, helping get these treatments forward faster. But before we start talking about news, I want to talk about numbers, because there is a lot about rare disease treatments that is about numbers. For example, all of you have heard this one. There are 7,000 rare diseases. That is a lot. CDKL5 deficiency is only one of those. So you have this giant swimming pool of balls, and we're just one. Imagine how hard it is for any individual ball to have enough attention, enough research, enough investment to have enough treatments. So my first question to you about numbers is, how many of the rare diseases do you think have a treatment approved? And I know that since this is a very smart community, you all follow the research field, probably half of you know this, but maybe for the other half, 5%, only 5% of all rare diseases have a treatment approved. So when you or your child get diagnosed with a rare disease, the most normal outcome is that the clinician says, and I am sorry, but there is no treatment for it. It's only 5% that have something. And 2022 was the year that we crossed the line and became part of the 5% club. The FDA already approved ganaxolone, the first treatment for CDKL5 deficiency disorder. We're waiting in Europe. EMA should also give them the green light, but that's towards the end of the year. And that is such a massive difference, because from now on, every family that gets diagnosed with CDKL5 deficiency will hear, and this is a rare disease. And by the way, there is a treatment approved. And again, that's just 5%. So this was such an important year for us. I'm going to ask you about a second number. And that is that, okay, ganaxolone is great, but it treats a symptom. It treats the epilepsy. And we know that the disease is so much more than that. How many rare diseases do you think have cures approved? And by cure, I mean something like a gene therapy, something that really corrects the root of the disease, the gene or the protein. How many? And I'm going to let you think about that number while I tell you about a story. And that's the story of another rare disease called AADC. This is aromatic amino acid decarboxylase deficiency. It's another enzyme in the brain. It's another deficiency that also leads to a neurodevelopmental disorder that is really complex. And we saw a lot about this last year because this drug was in phase 3 trials, this gene therapy. And this is a disorder where children are so hypotonic, so much. You can look at the picture at the very left of this girl, that they cannot do anything. They're just too soft. They also cannot speak. They also have intellectual disability, quite severe. It's just a global neurodevelopmental delay, but the hypotonia is amazing. And scientists have developed a virus where they take the DNA of the virus out and replace it by the DNA of the enzyme these kids are missing, AADC, so that when the virus is placed in the brain of these children, it will infect the neurons. It thinks it's infecting the neurons, but it's giving them the gene they need. And we saw the results of this trial. The results are really spectacular. At the CDKL5 forum, at the last one, we actually watched the videos, and you could see these children. This is the same girl 2 years after. She's walking by herself, and we could see them, some of them speaking, playing. They increase so much more their mobility and everything. This was a great therapy. And I'm telling you this because it just got approved in Europe. The EMA just gave the positive recommendation because EMA cannot approve. They just say, go ahead, and then the European Commission approves. But they just gave them that last month. And this is huge because this is another gene therapy to treat another rare genetic diseases that affects the brain, that is an enzyme that is missing. So very similar to CDKL5 deficiency. And it just got approved. And in the press releases, and here we go back to my question of the number, it says that this will be only the third gene therapy in the market. This is not 3%, 3 gene therapies in the market for all of the diseases, the rare and the not rare. This is how rare it is to have a curative treatment approved. As they explained, there are only two precedents. One is Luxturna, that's a gene therapy for an eye problem, retina. And the other one is Zolgensma, that's the gene therapy for SMA that is so famous. So I'm talking about the ones with the virus, the classical gene therapy, there are only three. It is so rare to have one because this is a field that is just starting in medicine. And sometimes we forget. And I think sometimes we think like, oh, everybody else has a gene therapy. Why is our disease not having a gene therapy yet? No, we're barely getting there in science. The first ones are coming out. And last month at the American Society for Cell and Gene Therapy, this is the biggest conference on gene therapies, there were 3 different gene therapies for CDKL5 deficiency presented. These are in mice, these haven't reached clinical trials, but I want you to think about the numbers. In the entire planet, there are only 3 diseases with a gene therapy. And there are so many thousands of diseases, and we have at least 3. I mean, I'm sure there are more, but we're just talking about the news that have been publicly announced, 3 at least in development. I am sure that in the near future, when we talk about having 2% of diseases, 3% with gene therapies, we are going to be there. We're going to beat the odds again, just like we joined the 5% club. And now let's talk about the news. That was the introduction. And I want to start by telling you a bit about the biology just to make sure we're all on the same page. CDKL5 deficiency disorder is a disease caused by deficiency in CDKL5. This is because the gene CDKL5 that is in the X chromosome, so girls we have 2 and males have only 1 of the copies is mutated. And if you're a male, well, you just have it mutated. And that mutation means that from that chromosome, the protein CDKL5 is the same name that is produced, is not going to work. It might be broken, it might be folded strange, it might be missing. The important thing is that we only have 1 good gene. So we only produce in half of the cells that good gene, and that is not enough. As a result of that, the brain cannot function well. I'm telling you this pathway because what I want you to know is that we have treatments in development for every single one of the steps. There are treatments in development to try to write the gene right so you can fix it, and now the brain can produce protein. There is also treatments in development similar to the one that I show you for AADC, that is the virus with the gene, that are gene therapies to give CDKL5 gene back to the brain. There are treatments in development to give CDKL5 protein already produced back to the brain. And we also have symptomatic treatments in development to help the brain function better and just make the symptoms less while we wait for all of these curative treatments to get there. And as these treatments move forward, they got to go through 4 different stages. The first one is in the lab. This is what happens with cells and test tubes, where scientists are thinking how to make the virus or how to make the medicine. It could be like the other medicines we have, a pill or a syrup that is going to fix the protein or the brain activity. So first part is designing it. Second part, we need to put that in a mouse that is deficient in CDKL5 so that we can confirm how well that treatment works. And at this stage, there's a lot of tweaking and fixing until it really works great in mice. Once it works great in mice that have CDKL5 deficiency, we take them forward to a 3rd stage that is preparation for clinical trials. This means scale up production because you need to make a lot of it so that you can go to clinical trials. It means figuring out the safe doses so you can go into trials with a safe dose, figuring out the distribution. With a pill or a medicine, normally it's easy, you just take it orally. But when you have a virus, you have to put it in some part of the brain, and that takes some work. So there might be some time also at this stage. Finally done, we can go to clinical trials. And right now what we have is symptomatic clinical trials with drugs for symptoms, and that's like ganaxolone that we just spoke about. But then we have treatments to fix the gene or the protein at every other stage, including the very last one right before clinical trials. So we have a full pipeline of things coming forward. And the most advanced ones is ganaxolone with this approval this year that made us part of the club of the 5%. And that approval has been in the US, Europe coming later. And the way that ganaxolone works, you know, is reducing seizures. There are several of these in treatment, in trials that we have that are all the same, reducing seizures, and it got the approval, which is the Happy New of the Year. There is a second one, also for epilepsy, that is also running a global study. And this is so important for a rare disease when you have to go to many countries and find at least 100 volunteers for the study. And this is fenfluramine from the company called Zogenix that now got bought by UCB Pharma, another big epilepsy company. And fenfluramine is already approved for 2 epilepsies, Dravet syndrome and Lennox-Gastaut syndrome. And they ran a very small clinical trial in CDD and it looked good. And so now it is in phase 3, international trials, recruiting right now. The requirements for this trial is that the child or adult has the diagnosis of CDD. They're looking for ages 1 to 35, so that's quite broad. You could be on the drug or you could be on placebo. That's what happens in these large phase 3 studies. And it also acts, as I was saying before, by reducing seizures. But this drug in Dravet syndrome is amazing. And I hope we get also some form of amazing results for CDD. This is not your average seizure drug. Then there is another global phase 3 study that is not really for CDD, but it's a potential opportunity for us. And let me explain you. This is another drug, in this case by the pharmaceutical Takeda, that also works by reducing seizures, but it does something strange to the cholesterol in the brain. It has a very unique way of working that makes it, to me, very interesting to try, in particular added to other drugs, because it's just so different how it does this thing. And Takeda ran phase 2 studies. Those are small clinical trials in 4 syndromes. One of those syndromes was CDD. And then now they've chosen to move to phase 3 to the global studies for 2 syndromes, Lennox-Gastaut and Dravet syndrome. The good thing is that Lennox-Gastaut is a diagnosis that is not based on genetics, it's based on the type of seizures and the EEG that the child has or the adult has. And there is a percentage of people with CDD that also have Lennox as the type of epilepsy. So for all of those, this can be yet another opportunity to get into a global study. They're looking for people ages 2 to 35, diagnosis of Lennox. You don't need any particular mutation. Just like the others, it could be drug or placebo, because that's how those trials work. But at the end of it, you get the drug anyway. And they're looking for a seizure type that Takeda has renamed. They call it major motor drop seizures. Those are not the classical drop, the ones that are just the head, but any seizure that results in falling. If you have at least 8 a month, you qualify for the study. If you have the diagnosis for Lennox, they're everywhere. Check out their website, see if they're near your country, ask your neurologist. So different options for phase 3s, and these are the symptomatic ones, but let's take a look at everything that is coming preclinical, preparing for the clinical trials that had announcements in the last 12 months. And the first big announcement in the last 12 months, and this was before Christmas, but it's just like the biggest announcement of the year, so I want to say it again, is the discovery that in CDD, it is never too late to come with the gene or the protein. And this was a discovery by Professor Zhou Zhao from the University of Pennsylvania, where he showed that he could make transgenic mice with a genetic switch on and off for the CDKL5 gene. So he made these mice be born with a switch off. So they're born not producing CDKL5. And he let them grow. So they develop without CDKL5. And they had CDKL5 deficiency disorder. They had all of the symptoms. And when they get to adulthood, he turns on the switch and lets CDKL5 starts being expressed. And at that point, he really, really had good efficacy. This led to a lot of press releases with the conclusion that CDKL5 is needed all the time. Not only at the beginning of development, and if you come later, it's too late. No, it's needed all the time. And because it's needed all the time, it is never too late to put CDKL5 back and see functional improvements. And this is something that has given so much energy to all of the gene therapies coming up and all of the curative treatments coming up for CDKL5 deficiency, because there was always that worry, oh, what if it is a neurodevelopmental disease and we've missed the good window? You don't. It is never too late. It's never too late in mice. There are simpler brains that we have in humans. We need to see in clinical trials, once we get there with the gene therapies, how much functional improvement we get, but we're going to get, because it is never too late. And this was the biggest discovery of the year, and probably is going to be the biggest of the decade. So let's go through all of those news in the last 12 months from all of these programs that are coming forward. The most advanced one is the one from Ultragenyx, from the ones that we have information. And this is a classical gene therapy. And by classical gene therapy, I mean like the one I told you before, you get a virus, you remove the DNA of the virus, you give it the CDKL5 gene, and now use it to put the gene back in the brain. And then those neurons have the gene and they can function well. This project by Ultragenyx is at the third stage of the steps that I show you. So they've gone through idea, they've gone through fixing the mice, and now they're preparing to scale up. They're preparing for those clinical trials. This is a lot of work. It's a lot easier to fix a mouse than to treat a patient. And at this conference last month, where there were so many announcements, Ultragenyx showed data of how they have now large-scale manufacturing of the gene therapy that they're going to be using for CDD. This works like at a brewery where you have these huge bioreactors, but instead of cells producing beer, is cells producing virus. So you can have a lot of viruses, and they're at that stage. And they're also working really hard on the clinical trial protocols and the data they need to get. And I think they have a presentation later. They're really, really preparing hard for those clinical trials to start at some point. Second project for which we have news this last year is the one from Amicus, and they're doing this in collaboration with Penn. This is a gene therapy with a twist. This is a gene therapy where they get a virus, they remove the DNA of the virus, and they replace it by the human CDKL5 gene, but they add a little sequence to that gene so that once CDKL5 protein is produced, it will also be exported around. And that's why I make this drawing. So you infect the first neuron, but it will produce protein, and just like a fountain, send it to everybody around. And that means that you will get a lot more in the brain than only with gene therapy. And they are at the stage of mice, figuring out how to get it to work really great in mice before they go forward. And they also presented data last month at this big American conference showing two things. One was this little fountain effect. They call it the cross correction, because you fix one and that one corrects the others around. But they also say the thing that is so important. And that is that in their experiments, they could see the remarkable possibility that the window for genetic therapeutic intervention in CDD is wider than previously recognized and may extend into adulthood. It means they're seeing the same thing that Joe was seeing with the mice with the genetic switch. You can come to adult mice, give them back CDKL5, and have functional improvements even in adults. So really, really cool results. Behind those, we still have more treatments. This one is from UC Davis that also presented last month at the big conference. What they're doing, it's also a type of gene therapy. This is like CRISPR, and I think you all know this. CRISPR is the cut and paste for DNA. This is a kind of CRISPR 2.0 that is targeted to opening up the second X chromosome. So if you remember, we have 2 copies of the gene in females, but each neuron is only going to read one. Some are going to be reading the good copy, but some are going to be reading the bad copy. And if you only read the bad copy, you have nothing else. You really miss the CDKL5. So what they're doing here is to open up the second one, so that by all neurons expressing both copies, one is mutated, one is fine, all neurons are fine because they all have one copy, and one copy is all you need. So this is the approach. It will be for females. And they are also in that mouse stage where they're figuring out how to make it work well and tweaking before they can move forward. And they presented that last month, how they're using their gene therapy, because this is a type of gene therapy, to reactivate the second copy. And what they're showing is that the reactivation, they still haven't advanced enough to show all the efficacy and the safety, but they are at that stage. They're working in mice every year moving forward. This is the group from Kyle Fink. Another very smart team at another famous university in Harvard is David Liu team. What they're doing is also a version of CRISPR, but in this case is the one that will rewrite the DNA and fix the gene. So what they're going to do is not to open the other copy, but to fix the wrong copy. And they're doing that using this type of CRISPR. And what they've published this year, and this was a publication in one of the biggest journals, Possible Cell, is that they can fix cells in vitro from a girl with CDD. And this girl with CDD has a very tricky mutation, because some of the mutations in CDKL5 are that one of your DNA letters is changed by another letter. And that could be a missense, or it could be a nonsense. These are the main type of mutations. But sometimes it's not that you got the wrong letter, it's that you're really missing a letter. And that's a lot harder to fix, because you cannot just rewrite that letter, you have to insert. And this is what this advanced CRISPR technique is doing. In vitro, in cells from this girl, it can find the wrong CDKL5, the one that is missing one letter, and find that spot and put the good letter. And this looks like science fiction, but they're already doing it in cells. So in that first step, so if we step back again and look at all of the things that are coming up, we have those clinical trials with symptomatic treatments where we have the first treatment approved. We're in the club of 5%. That's my big slogan for this year meeting by Marinus. Marinus is expecting soon approval in other territories. And in the meantime, they have an expanded access program. It's different depending on the country so that they can make it easy to access ganaxolone while we wait for it to be available through their health system. Second clinical trial, global, Zogenix with fenfluramine, very promising drug. US, Europe, many places, check out the website. Takeda, not exactly for CDD, but for Lennox, early data in CDD from trials. See if you can also consider this one through your centers. All of these are for symptom, epilepsy. They might have other symptoms, but it's essentially a symptomatic treatment. So other symptoms that it helps. In clinical trials, but behind that we have all of these other treatments. We have classical gene therapy, the most advanced that we have news, the one from Ultragenyx. Behind that in mice, we have this gene and protein therapy combo from Amicus. We have the one going after the X3 activation that is really cool because you have that second copy sitting there. If only you could use it. And then we also have the most advanced forms of gene editing that are gonna fix the DNA, and then you're ready to go because your DNA is already normal. An important message that I have for you is that these are the news and announcements, and this is just the tip of the iceberg. There's so much more going on around CDD and treatments for CDD, but that haven't been announced. So this is really an iceberg. And we happen to know many of those because from the LouLou Foundation, we're exposed to many of this. We launched recently an observational study, so to learn about symptoms, worldwide, 100 patients, and that study is in collaboration with 7 companies. And 6 of those are public companies, big companies. It's hard for them to put their money on a disease, and they're putting it in CDD. So there is that interest, and there are activities, but things that are not in the list of announcements that I have here today. We also are in constant dialogue with more than 20 different companies. They're following their space, or they have early research in the space. So my message is there is a lot more, and it is important that there is a lot more. Because science is hard and many of these projects may fail, and business is hard. And right now, if you know the markets are going down and companies might then need to give someone else the program so that it's easy, it gets forward. So science is hard, business is hard. What we need to have is many working on it, and we have many working on CDD. So that was the bulk of my presentation, just a summary news since the last Alliance meeting. Now that we're in beautiful Canada, I wish I was there, I really love Canada, is first drug approved for CDD. We've joined the 5% club. This is big, this is big. Only 5% of rare diseases have this. Second global phase 3 trial with fenfluramine, potential to get into another phase 3 with soticlestat. This is the year we learn, the last 12 months that we learn that CDD is largely reversible in mice, that it is never too late. Looking forward to get there with those gene therapies. And by the way, we have so many multiple treatments to fix the gene or fix the protein, advancing towards trials. Every year, we see them all moving forward, one box. We're gonna get there, we're gonna make it. And you have so many other news in this conference after me. You're gonna hear about registries, outcome measures, biomarkers, other programs. I hope you all enjoy the update and that you're pumped and hopeful, and I look forward to your questions now. I'll stop sharing.